Good morning. My name is Thomas O'Connor, and I'm the Director of the Division of Product Quality Research and a member of the Emerging Technology team. Today, I'll be talking about fostering innovation through OPQ's Emerging Technology program. First, I'll briefly describe the Emerging Technology program and the Emerging Technology team, then highlight some of the emerging technologies we've encountered through the program. Finally, conclude with a couple of case studies that illustrate how collaboration through the Emerging Technology program can support the implementation of innovations in pharmaceutical development and manufacturing. Why emerging technology is important for both the FDA and the pharmaceutical industry. We feel innovations is an important component of addressing challenges in pharmaceutical development and manufacturing. First, it can help address some of the underlying causes of drug shortages and help mitigate or prevent future production problems by addressing the root causes of product failures. Provide a flexible and agile manufacturing sector by improving manufacturing efficiency, increasing process robustness, lowering manufacturing costs, increasing supply chain flexibility. Finally, novel analytical tools can facilitate the development of complex products, improving the availability of complex generic products to the American public. The Emerging Technology program is open to both new drugs as well as generic drugs. And CDER's Emerging Technology team is already working with several generic sponsors to support them as they work to implement innovations for their pharmaceutical development and manufacturing of the products they make. There are challenges to implementation, though. Adoption of emerging technology can require new investment. It has to be applicable to the changing inventory of products that are being manufactured by a generic manufacturer, and the development of new knowledge and skills. We feel, though, that through collaboration, we can overcome these challenges and foster innovation. The Emerging Technology program was formed to encourage and support the adoption of innovative technology in pharmaceutical development and manufacturing, with a focus on close collaboration with industry and other stakeholders, realizing that we need to work together in order to make this happen. The heart of the program is the Emerging Technology team, which is a small cross-functional team with representatives from all the FDA quality assessment inspection programs, including OPQ, Office of Compliance, and ORA. The objectives of the program is to first serve as a centralized location for external inquiries on novel technologies, providing that single point of contact. Then the other unique part of the program is early engagement. It provides an opportunity for sponsors to engage early with the FDA as they look to implement innovative solutions and technologies in their pharmaceutical development and manufacturing. And this early engagement can occur even before a specific product is identified, which is a unique aspect of this program. Then, to ensure consistency and continuity and predictability in the review and inspection by following that early engagement all the way through the application and eventual approval process. Another aspect of the program is to engage with other stakeholders, including international regulatory agencies, to support global adoption of innovative technologies. Also, this early engagement allows us to identify and evaluate potential roadblocks relating to existing guidance, policy, or practice, and be proactive about addressing those before the application is submitted. Finally, as an innovative technology or emerging technology matures, the Emerging Technology team can help facilitate knowledge transfer to other CDER and ORA review and inspection programs, and to help establish scientific standards and policy as needed. As I mentioned, the focus of the Emerging Technology team is really on collaboration, and that collaboration starts in the early engagement space. That early engagement can take the form of a Type C meeting, but it can also include emerging technology site visits, where the FDA can come visit the site where the innovative technology will be implemented, to really have more prolonged engagements that facilitate shared learning and get the facility and the application ready to be submitted. Finally, that same ETT representative will follow the application throughout the entire process, providing that continuity. And that means participating in the integrated quality assessment, and maybe even attending the pre-approval inspection. We try to form the composition of the review team as early as possible, and maintain that team throughout the entire process. To learn more about the Emergent Technology Team and the Emergent Technology Program, please visit the website listed here on FDA's website. Next, I wanted to just share kind of what we consider emerging technologies as part of this program. First, it can be a novel manufacturing method to improve process robustness and efficiency. An example here could be continuous manufacturing. But it can also be novel dosage forms or delivery systems intended to improve drug delivery or targeting. And we've encountered examples. Listed shown here is a 3D printed tablet, but we've encountered other novel dosage forms through the program. It can also include novel analytical tools intended to improve product quality, testing, process monitoring, and/or control. The common thing between all the technologies we've encountered through the program is that FDA has limited assessment experience due to its relative novelty in the pharmaceutical industry, and that the technology has a potential impact on product quality. It's these two factors, novelty and potential impact on product quality, that we evaluate when considering submissions to the Emerging Technology Program. What are some examples of emerging technologies that sponsors have brought forth to be part of the program? Certainly continuous manufacturing has been an active area, but it's been more than just continuous manufacturing and includes some novel control strategy approaches, both based on modeling and novel analytical approaches, as well as novel dosage forms. Another active area has been in the manufacturing of sterile products. And a lot of innovations here relate to both the monitoring and the testing of those products, so new analytical approaches, as well as the manufacturing process itself. Innovations in the aseptic filling process, robotics, and even the novel container closure systems. Finally, I want to conclude my talk with just highlighting a couple case studies that highlight how the innovations can impact public health. So the first case study centers around a novel analytical approach. In vitro release tests have increasingly become important in product development as well as regulatory assessment of complex products, such as nanoemulsions, suspensions, multivicular liposomes, and microspheres, as it provides key information about the quality and performance of drug products. An ideal IVRT method can correlate the changes in critical quality attributes of the formulation directly to its drug release characteristics, and thus provide valuable information to ensure batch-to-batch consistency and quality, allowing determination and assessment of post-approval changes, and allow product comparison to support the determination of bioequivalence. Most of the currently available IVRT methods, such as microdialysis, reverse dialysis, focus on quantifying the extent of drug release, and to a lesser extent, on the rate of drug release. Furthermore, these commonly used IVRT methods generally rely on the diffusion process via the dialysis membrane to separate the released drug from the remaining one, which has an inherent limitation. First, the use of membranes invariably introduces a rate-limiting diffusion step, and thus prolongs the overall duration of the test. This may not be suitable for formulations or routes of administrations that have rapid drug release characteristics, nor would it be sensitive enough to discern minor differences in drug release from complex formulations. To address these challenges, a novel in vitro release test method was developed called adaptive perfusion. Adaptive perfusion is a pressure-driven separation method based on the principles of tangential flow filtration. The unique features of adaptive perfusion method is its ability to achieve size-based separation of the particulates with simultaneous analysis of the released drug as well as the remaining drug. In comparison to the conventional dialysis method, the adaptive perfusion method can discern the rate and extent of drug release from drug solution, drug-loaded micelles, and nanoemulsions via the control of several process parameters, such as the filter molecular weight cutoff, feed flow rate, and back pressure. The adaptive perfusion method provides discriminatory drug release profiles for drugs in solution, micelles, and small and medium large globule size nanoemulsions. Compared to the dialysis method, a significantly faster, i.e. minutes rather than hours, and greater release was achieved with this method. Shown in the figure here is an example of such results. The overlay shows the in vitro drug release profile for small, large globule size nanoemulsions tested using adaptive perfusion compared to a dialysis method, which shows both the quicker test as well as the discriminatory power of the test. These novel approaches can support the availability of complex generic products to the American public and thus provide a public health benefit. For more details on the method, you can please reference the paper listed here below. The second case study I would like to highlight is quality attributes of the container closure system for parenterals. Pharmaceutical containers for parenterals have been predominantly manufactured using glass as a packaging material choice, especially type 1 glass, since it's regarded as chemically inert and an effective container closure system. Nonetheless, there have been reports and recalls related to glass quality issues, such as breakage, flakes, particles observed in marketed products. Under certain conditions, glass has the potential to shed fragments, called glass laminal or flakes, glass particles, and metal extractables. There have been recalls due to glass flakes, breakage, and particles observed in marketed formulations. And a couple of examples are listed here. Based on literature reports, borosilicate glass vials may undergo the dissolution of the glass network at high pH or elevated temperatures, producing silicate particles or flakes. Other common problems observed in glass containers are breakage, cracks, chipping, abrasion, and particulates generated due to stresses encountered during the product manufacturing, the filling process, transportation, or even during administration. And these product failures can lead to safety issues. Through the Emerging Technology Program, sponsors have brought forth innovative container closure systems that hope to address some of these challenges. FDA has developed a systematic platform to test approaches capable of assessing common failure modes of pharmaceutical glass and container closure systems. Notably, they focused on two types of failure modes: mechanical stresses, the breakage and cracks, as well as chemical stresses, particulate generation, metal leaching, etc. Applying orthogonal analytical methods to detect the extent of the failure mode and potential impact on product quality. And this includes like freeze-thawing and other analysis. The comprehensive assessment of pharmaceutical glass containers requires evaluation of the physical properties of the glass, understanding the chemical interactions between the glass and the drug product, and understanding the mechanical properties of the glass container. These scientific approaches were able then to inform the regulatory assessment of applications using these novel container closure systems. And through the Emerging Technology Program, we actually even collaborated on filing strategies to speed adoption across product lines and manufacturing sites. And the product health impact of this was increased supply of high-quality vials for injectable drug products and vaccines. Here's a couple of papers listed below that describe in more detail the comprehensive analytical approaches that were used to evaluate these novel innovation in container closure systems. Finally, I just wanted to conclude by highlighting that together we can work to overcome challenges and foster implementation of innovations that improve product quality. And we really invite sponsors who are looking to implement innovations in their pharmaceutical development and manufacturing to reach out to the Emerging Technology Program if they have questions about especially how this would fit within the current regulatory assessment programs. Finally, I'd like to conclude with a couple of challenge questions. Is this statement true or false? Emerging technology provide benefits to the generic drug industry. This is true, and the emerging technology team is already working with a number of generic drug sponsors to help support them as they look to implement innovations in their pharmaceutical development and manufacturing. Collaborative interactions with the emerging technology team can help navigate potential regulatory uncertainty when implementing novel technology. This is true. Through the early engagement, we can proactively identify regulatory challenges and work to address them even before the application is filed, eliminating that regulatory uncertainty. With that, I would like to conclude by thanking all the members of the emerging technology team. OPQ leadership, and most importantly, our industry sponsors who have been bringing these innovations forward and are looking and have been great partners through the program. Thank you for your time.